Hello friends, welcome to my channel Bioknowledge with DhanaShri. Today our topic is History of Microbiology Part 1. I have divided History of Microbiology in 3 parts. So I'm going to tell you Part 1 in this video lecture. So let's get into the video. History of Microbiology. Yes, microbiology means what? Microbiology is the study of small living organism. It's a specialized area of biology that deals with the living things ordinarily too small to be seen without magnification. Yes, it need to be magnify that particular microorganism. Yes, for the magnification that microscope should be there. So such microscopic organisms are collectively called as microorganism or microbe. Just microorganisms and microbe. You can take these words. Okay, so both are same, microorganism and microbe. So microbiology started after the invention of true microscope and the discovery of microorganism. So Roger Bacon in 1267 for the first time described lens. He postulated that disease is produced by invisible living creature. This suggestion was made again by Fracastoro of Verona and Anton von Plancius in 1762, but these people had no proof. So Zacharias Janssen in 1590, the Dutch spectacle maker for the first time produced crude microscope by placing two lenses together without any provision focusing lens. So the modern microscope are based on the same principle. Okay, Galileo Galilei in 1610 invented true telescope microscope, but its resolving power was very low. So he observed water flea which appeared to be as larger as hen. So Athanasius Kircher, again another scientist, in 1658 observed plague bacilli from blood of a person suffering from plague, and he gave the name as minute worm. The magnification of microscope used by him is only 32x, and evidently he mistook RBC as plague bacilli. Okay, he mistook the RBC. He thought that is a plague bacilli, but it was a RBC. So he referred to worms, yes, invisible to the naked eye in decaying bodies, meat, milk and aerial secretions. Although his description lacked accuracy actually. So Kircher was the first person to recognize the significance of bacteria and other microbes in disease. Again, another scientist, Fracastoro of Verona, published a book on contagious diseases, and in his book he reported that agent of communicable disease cause contagium vivum, that is minute particle. So he put forward the idea that infection due to minute particles and disease are spread by his particles. He said that some minute particles are there and they are spreading the disease. So he gave the name to this minute particle as Seminaria marbi, too small to be seen with the naked eye. That meaning of that is too small, very small, so that we can't see by naked eye. So he further defined infection and infectious disease that were transmitted per contact, direct contact or per fomites or at distance, means through air also. And again another scientist, Robert Hooke, yes, this is a very famous name, that he gave the name cell. So in 1665 saw and described cell in a piece of cork. Okay, he used the word cell for the first time. Yes, whatever we using the cell at the present, that is described by the Robert Hooke scientist. He described the structure of plant cell. He observed honeycomb like structure in a thin slice of cork which was surrounded by a cell wall. He wrote a book called Micrographia when he made all his observation. So when he made all his observation, he wrote a book that called as a Micrographia. Okay, and he made all observation in that book. So he described the cell in a piece of cork, established that fact the bodies of animals and plants complex, okay, they are complex as they may appear, are yet composed of few elementary parts frequently repeated. Okay, a quotation from Hooke, not from Hooke but from Aristotle description of the cellular structure of living things. So although he was not first to see the bacteria and protozoa, here the name Antony van Leeuwenhoek was the first person to report description of microorganism in detail. Okay, the discovery of microbiology as a discipline could be traced along the following historical areas. What are those era? Discovery era, transition era, golden era, and the era of molecular biology discovery in 20th century. Okay, so in this video we'll see the discovery era. Okay, after that in further videos, next videos, upcoming videos, we'll see the transition era, golden era, and era of molecular biology discovery. These the history. Why we need to study the history? Because how they discovered the microorganism, how that subject is evolved. Yes, we need to understand that before going detail into the microbiology subject. So first we'll see the discovery era. So here you can era discovery era, this period concerned with the discovery of microbial world that has been dominated by Antony van Leeuwenhoek. Yes, Antony van Leeuwenhoek 1632 to 1723. So the fundamental observation in microbiology started from the invention of true microscope by Antony van Leeuwenhoek in the last quarter of 17th century and the early part of 18th century. Means it is a transition period between 17th and 18th century. So he was a cloth merchant living in Delft, Holland. He earned his living as a draper and haberdasher, a dealer in men's clothing and accessories. So he had little education but great patience and curiosity. It was his hobby to prepare lenses by grinding the glasses. It was actually his hobby. Okay, by that hobby we got the microscope now. So he spent much of his spare time constructing simple microscope composed of double convex glass lenses held between two silver plates. He produced very simple type of microscope having the magnification about 40x to 300x consisting of home ground lenses. So the compound microscope of today having magnification from 100x to 3000x are based on the same principle of Leeuwenhoek simple microscope. Yes, you can see here the Van Leeuwenhoek using his microscope. How small it is, you can see with the hand. He put like this and he's seeing, he's observing. So during his lifetime, he made more than 250 microscope consisting of home ground lenses mounted in brass and silver. Okay, the most powerful of which could magnify about 200 to 300 times, which allow him to observe microscopic algae, protozoa and other larger bacteria. The lenses of Leeuwenhoek microscope were well made and Leeuwenhoek had the openness of mind that much very important to an investigator. His description of protozoa were so accurate, okay, so accurate that many of the forms he described are easily recognized today also. So in Leeuwenhoek simple microscope, specimen were placed on the point and adjusted so that lay in front of the tiny lens. You can see here what are the parts of that particular microscope, Leeuwenhoek microscope. Lens here, specimen holder, it is specimen holder, and focus screw, this is, yes, you can focus that and focus the particular specimen, and it is a handle to hold the microscope. So specimen was placed so that the beam of sunlight from here, okay, passed through it and viewer looked to the tiny lens at the illuminated material. So thus Leeuwenhoek was the inventor of true microscope. Therefore he is known as father of microbiology. Okay. So with the help of his simple microscope, he observed minute organism in rainwater, seawater, and scraping from teeth. He gave the name little animalcule to this minute organism. So whatever he see the small organism, he gave the name little animalcules. He made the accurate description and sketching of various bacteria he observed. Whatever he observed, he given the sketches of that bacteria. He draw the structure of that particular bacteria, whatever he seen with his simple microscope. So some of the bacteria were rod shaped, bend like sticks, spiral in shape, and some were of spherical shape. Some of the bacteria were actively motile also. So here He communicated all his results of his experiment to the Royal Society of London in 1676. So whatever he got the results, okay, from his from the microscope, like whatever he see the microscopic organism, he communicated all his results to the Royal Society of London in 1676. So thus Leeuwenhoek was the first greatest of all microbiologists because he discovered bacteria, protozoa, spirochetes from mouth, intestine and water. So thus Leeuwenhoek discovered true bacterial and microbial world. Therefore he is known as father of microbiology, father of bacteriology and protozoology. So this is all about the discovery era in the history of microbiology. In upcoming videos, we will see transition era and golden era. So thanks for watching this video. Stay tuned for more videos. Thank you.